Okay, I see some people trickling in. Good morning, everyone. We'll go ahead and get started. We only have a half an hour planned for this webinar. Can everyone hear me okay? Yes, perfect. Thank you for joining us this morning. Well, morning for me and morning for Lilian might not be morning for everyone. So good morning, good afternoon, or good evening, wherever you are located. I'm Randee Hunter. I'm the current chair of the Scientific Leadership Committee for the International Society of Bone Morphometry, and I'll let Lilian go ahead. You can introduce yourself.
I am Lilian Plotkin. I'm a former member of the board, and I'm a professor in Vienna University School of Medicine.
And Lillian and I are here this morning at the request of the Early Career Committee for ISBM. They thought it would be a good idea for us to just give some tips and tricks for writing an amazing abstract for the upcoming 2026 meeting of ISBM in July. So the plan for today is just to give you a little bit of an overview if you're new here or if you don't have a lot of experience with what ISBM is. We'll go over a little bit of our scope of interest, and I think that will help with guiding you towards the types of abstracts that you might be thinking about writing to submit to the meeting. A basic introduction of the abstract guidelines, where to find them, what we're looking at, what we're expecting, and some tips for writing an amazing abstract. And then we'll actually do a little bit of a Q&A dissection of an amazing abstract that Lillian has provided for us, just to look at some of the components that we're looking for as reviewers of ISBM abstracts for 2026. And at the end, we'll have time for some open questions, but also during this process, on any of the slides and any of the content, please feel free to raise your hand and ask questions. This doesn't just have to be us talking to you. We'd like for it to be interactive, and this is hopefully going to be a tool for you all to get more comfortable with writing an abstract to submit for ISBM. So just to give you a little bit of background, the International Society of Bone Morphometry is really interested in quantitative analysis of form and function of bone across any scale. So we're talking whole bone, whole human, all the way down to cellular and nanostructure scale. A lot of the work that has been concentrated on in our society is to advance the different imaging and visualization techniques of bone, and really to help this collaboration between clinical therapeutics and research. As you'll see at the bottom there, that's a direct quote from a recent paper in 2024, which is linked at the top. If you're interested in some information about the history of the International Society of Bone Morphometry, many of our past presidents and current president worked together to outline sort of here's how we got to where we're at now, and here are some of the influential players. But on the bottom quote there, you can see the ISBM is dedicated to advancing research, education, and clinical practice for osteoporosis and other bone disorders by developing and improving tools for quantitative imaging and analysis of bone. So a lot of our presentations at ISBM Congress, which occurs every 2 years, are really geared towards how can we move the field forward in terms of quantifying aspects about bone. And so we see a lot of new novel techniques, novel applications of existing techniques to be able to visualize and quantify aspects or traits of bone. Some basic information. I assume that you are here because you already have this on your radar, but our next ISBM Congress is in London from July 4th to July 7th. It corresponds with the British Bone Society's meeting as well. Some of the basic aspects of what we're looking for in your abstract submission are similar to other meetings and programs that you may have submitted to. We're looking for an abstract title. We'd like all your co-authors and their affiliations to be listed, including for you to indicate the presenting author. Our abstract content is 2,500 characters. That does not include spaces. This aligns with some of the other meetings that you may or may not have submitted to. For example, the American Society of Bone and Mineral Research also requires the same length. We ask that those abstracts are in English for our reviewers, that they are clearly organized with a clearly stated purpose, materials and methods, results and conclusions of the data that you are presenting, that you give us sufficient detail as reviewers to assess the scientific rigor of your processes, your data analysis, as well as interpreting your conclusions. And you are allowed one captioned figure or table. We can talk about this in the next slide. You do not have to submit a figure or a table, but that is allowed in the submission. Our deadline for our normal deadline for abstracts is March 1st, 2026. This is for anyone who's interested in doing either a poster presentation or an oral presentation. All abstracts will be evaluated for either a poster or an oral presentation. We do not have you designate whether or not you'd like either one. We do have a late breaking deadline at the end of April, April 30th, 2026. That will be only for posters. We will fill our oral content, our oral slots in our program from the March 1st deadline submissions. I think I have a question in the chat.
It's just a link
no,
to the
just a
website.
link. Thanks. Thank you. Thank you, Jason. And we can share out these slides as well that do have active links. So these abstract guidelines that Jason put in the chat are linked here. We are not going to talk today about our travel awards. There are 4 awards on our website that you can look at the eligibility and requirements. If you have questions about those, we're happy to answer those at the end, but we're not going to go into much detail about those now. This is mainly focused on the abstracts. Your abstract submission, if you are interested in also supplying the additional materials that are listed on the website for awards, that must be by the March 1st deadline. If you use the late breaking deadline, we will not consider those eligible for any of our travel awards. And then lastly, the submission to ISBM of your abstract will be emailing the email linked here, isbmsociety@gmail.com. Okay, so let's go into a little bit of detail about what makes an amazing abstract. Really what we're looking for is for you to demonstrate to us in 2,500 characters the scientific rigor of your work. So giving a brief background or context, clearly stating the purpose of your work. This should be only a few sentences at the beginning. The bulk of your abstract should be dedicated to providing us with information about transparent methods, materials, and then reporting your results. It is okay if you are doing some preliminary work. It doesn't have to be the entirety of your project, but we do need you to have a transparent and detailed explanation of your materials and methods. The brief interpretation, summary, or application at the end of your abstract is always good to tie this together, especially if you have a lot of details in your results. We want to see that you can bring it back out to what is the application, particularly to an audience for ISBM that's interested in sort of moving that, moving the field of bone research forward to improve clinical applications. That does not necessarily mean that you have to have clinical applications of the data that you're presenting, but we do have to see that sort of in the context of the larger picture. And that's what that fit for ISBM or the fit for our scope. Remember that we're looking at quantitative analysis of bone morphometry, but that morphometry can be at any scale. The use of the figure or the table, use those to demonstrate any methods, demonstrate new ways that you're visualizing bone. Use those to demonstrate some data that maybe was too complex to include in only 2,500 characters with the other expectations. Help summarize those data or help to interpret your results. Again, it is not necessary for you to include a figure, but see it as an opportunity to help the reviewers really dig into what you're presenting. Okay, I will stop there with just sort of the brief tips and tricks and basic outline for what you need for the abstract. Are there any questions at this moment before we go through a little bit of an exercise? Or anything, Lillian, that you'd like to add that I missed here?
I would emphasize that you have to make sure that you're tailoring your abstract to your audience. So if you're sending an abstract that is associated with a disease to a general bone meeting, make sure that people that will review the abstract understand what the disease is and why that could be important. And make sure, I mean, the best abstract always have data, numbers, not just it went up or went down sort of things. So make sure that the reviewers can appreciate the differences and why you're saying that the things are different.
Yeah, I think that transparency in both how you obtain the data, how you analyze the data, and what those results are is really critical for reviewers to essentially trust what you're telling us is happening with your data. Any questions at this stage?
I can ask one. If we are developing a method, do you consider that the validation steps that we have all along the development of these methods are results? Or is it something that like we explain that we have developed this method and then show the results that we got once we have developed the method? What would the validation steps that can include quantification, right, be also results that would be receivable in an abstract for the ISBM? Or
Go ahead, Lillian.
Yeah, and I think this is what we are doing. These are your results, even though it will end up being a validation of a method. I think that should be included as results. And you could have just that and end up with a conclusion that this method can be used, or you can apply it immediately to something else. But anything that you are doing, it should be considered as result, I think. I don't know, Randee, you have any?
Yeah, I completely agree. And I think one of the strengths of ISBM Congress and our at our meetings is a lot of the networking and a lot of what we end up talking about is sort of that aspect as well. We're trying to share these new and novel techniques with each other. And so I think that that's a completely appropriate abstract to submit and that you would want to include those quantitative pieces of the validation.
thank you.
I have one question in the chat from Lisbeth. Should you have headers for each paragraph, background methods? That is a stylistic preference. And so if that helps you to organize your content, that is completely fine. But we don't have a sort of structured template like that.
And I think that's how you should think about your abstract, but that is going to count against your limit of characters or words. So I mean, unless you don't have a lot to say in the abstract, I would avoid those to just be able to explain better what you're doing or get more information on the results.
Yes. Okay, seeing no other questions at the moment, I will turn it over to Lillian for our exercise or our example, and I'll let her go through this with you.
Okay, so this is an ASBMR abstract, and it was my first ASBMR abstract when I moved to the US. I will tell you later the year, but so this was a novel idea, and so we have to start by saying why we are doing this. And so the first two sentences, the first one is setting up the stage, why we think that we need to do these experiments. And I'm sorry, my mouse is not working, so I cannot point out. So if okay, so and this, then the second sentence, again, we're saying why we thought that we needed to study the biphosphonates on the effects of glucocorticoid-induced bone loss. And just to put you into perspective, at this time, the thought was biphosphonates were anti-resorptive, so they will kill osteoclast, they will inhibit osteoclast activity. And we were showing for the first time that they were having a different function and different effect. So the third sentence is the methods. So we were using this cell line. We transduced them with a green fluorescent protein targeted to the nucleus. And that's what we are using for the experiments. And we use these cells because with this transfection, we can see the nucleus. And we can see the shape of the nucleus. And so we go to the treatments. We treat them 1 hour with vehicle or the concentration of biphosphonates that we use, and we use different biphosphonates. And then we add the glucocorticoids. After incubation, we count the cells, and we selected 500 cells randomly. So then we go to the controls. Only 7% of the vehicle-treated cells exhibited apoptotic features, whereas 24% of the cells with dexamethasone exhibit the apoptosis. So this is our control. And then we went to the effects of biphosphonates. So one criticism I will do to this abstract is that we don't have standard deviations. We just have means. And so we confirm the results using a different method, and that's the Trape and Blue uptake. And then we did those dependent effects. So that's our results. And then we go to the conclusion. We didn't know at this point how the biphosphonates would reach osteocytes in the mechanism. And that was what it took probably 10 more years of my career trying to figure out. And then an idea that we still think it's valid, that the anti-fracture properties of biphosphonates may be due in part to anti-apoptotic effects on osteocytes. So that's the conclusion of the abstract. So this abstract was submitted in 1998 and was accepted for oral presentation. And at that point was a joint meeting of the ISBM and the International Bone Society. So any questions, comments, any criticism, anything that you think it could be done better? And this is a purely in vitro study. And again, it was a long time ago, so it may not fly right now. When we want more mechanistic studies and more sophisticated methods, this was very simple.
Lilian,
Yes.
you mentioned the fact earlier that we had to adapt the abstract to the audience.
Yes.
How do we know who is the audience? I mean, who is evaluating the abstracts at the ISBM? How does it work when we are new to it and we don't know what's the process?
So I will look at the mission of the society and what Randee talked about, what the interest of the society is. I have two hearts. one is the bone field and the other one is the connexing field. So I write abstracts for both bone societies or bone-related meetings, and also for the connexing meeting. So if I'm writing an abstract for a connexing meeting, I have to tell them what an osteocyte is and why I'm focusing on osteocytes. If you're thinking about a bone meeting, you don't need to do that because everybody knows what an osteocyte is. So that's what I'm thinking when I say you have to tailor to the meeting and to the audience. Make sure that there's no, I don't like acronyms, and I think it gets confusing. I know that we need to use them for because the limit of words that or the characters that we have to use, but if you're using acronyms that are very well known in one field, they may not be known in the other one. It may mean something different, so make sure that you spell out things. If I'm using BMD for a bond meeting, I'm probably not going to spell it out. But if I'm sending something to a connexing meeting, yes, I'm going to say what BMD is.
Yeah, and I'll just add, it doesn't necessarily matter the backgrounds of all the reviewers of the abstracts because we're all guided by the same principle that Lilian mentioned, we talked about earlier, that we're really looking for innovative science that applies to the mission of ISBM. And that will be, interesting conversations at ISBM Congress meeting with each other. Which I think is the strength of our meeting, is that there's so much opportunity for us to network with each other and discuss the science that we're doing. So the critical pieces are that the way in which you present your work in the abstract is clear for someone who might do maybe HRPQCT work, but is reviewing something that is more on the cellular or spatial omics level. So that being able to clearly communicate your science, I think, is sort of a guiding principle no matter where you're submitting. But that we can see the scientific rigor and the tie to the mission of ISBM is really the most important part beyond, the who is actually reviewing the abstracts.
Question in the chat.
Yes, Lisbeth, it is possible. It is optional to add a figure or a table, but it is allowed. And as I mentioned, I think if that figure or abstract helps the authors to summarize data, present data that maybe the word limit or the character limit doesn't allow you to potentially elaborate further on, or if it makes a concept about data collection or data results more clear, then I think you should definitely use that. Lilian, do you have other tips on that?
No, I think that a graphic abstract will be a good addition because that helps also put together in your head what you mean by your abstract, so and will help the reviewers understand that, and then ultimately help the people that attend the meeting to know what your thoughts are. But yeah, you can use it at your advantage whenever, whatever way you like.
Let's see. Okay, how much detail would you go into stating the research gap that is being addressed? In my opinion, and then I'll throw it to Lilian, really, Shivani, the first one or two sentences should sort of outline the research gap or what you're talking about. So as Lilian mentioned before, if you're talking about a particular bone disease, that might be something that you want to sort of tie together with the research gap. But being that this is a limited abstract, we have to know why this work that you're doing is important, but without sacrificing too much space or characters to tell us or show us how you're addressing this.
Yeah, I agree. Not more than 2 sentences, an introduction, and then go to what you did, the methods.
Mhm.
It's ideal.
Agree. Historically, what percentage of submissions utilize a figure? I don't know that I have those data. I'm not sure. I'm sorry, Ingrid. I don't know the answer to that question.
And this is a relatively new thing. So I don't know for how many years we have been having images added to the abstract. So
Mhm.
it would be interesting to know. Yeah, I agree. But I think it's, as I said, you should use it to your advantage and try to convey either if your methods are complicated, have a scheme of the methods, or if you have a conclusion that will benefit with an image, that always helps.
Any other questions? We have a few minutes left. I'll go ahead and put the information slide back up. I actually think that I have a typo here. I apologize for that. Jason, can you confirm, is that ISBM Society without a period in there?
There's a period.
Oh,
Should be
there
a
is.
period.
Okay. I
Yeah,
was right the first time. Good. Okay.
Yeah.
ISBM.society.
And can you confirm there is a LinkedIn page where people can continue the conversation?
Yes, so I'll post this recording of the webinar to the LinkedIn page, and then people can ask questions. I'll put a note that they can ask questions.
And ideally, it could be useful for you to discuss among yourselves, and because I mean, at least myself, I have a standard way of doing things, and then sort of set after so many years. But you may find by talking to each other that you find a better way to express what you want to convey in the abstract. So I think that the conversation is also important.
Mhm. Elizabeth asked if we could talk a little bit about what's important when scoring the abstracts. Excuse me. Yeah, I think kind of what we've covered here, we want to trust the science that's being presented. So the transparency in methods, the rigor, the actual quantitative results being reported in the abstract, that's a general, if you're submitting anywhere, kind of best practices for the reviewers to be able to judge the scientific rigor of what you're doing. And then the other pieces are for the abstract to fit into what we're looking for at ISBM, whether it's appropriate for our meeting. Is it meeting that mission of ISBM? And then can you sort of tie it up with a bow and tell us what this could be, what this application, or sorry, what these results, the application of that could be. In my opinion, those are kind of the big buckets that the reviewers are looking at in terms of scoring.
Yeah, I also like to see a story when I read an abstract, and that is not taking me a lot of time to go through and have to fight to understand.
Mm-hmm. Yeah,
Yeah.
clarity in the communication of your science is really key. We will be reviewing as reviewers multiple abstracts. So as Lilian mentioned, if we're having a hard time understanding what you're presenting, that makes it more difficult for the score to actually reflect potentially the work that you're presenting.
And it's not a bad practice to ask somebody that is not directly related with the work to read it and comment on that, because people that are going to review are not people that are in your lab. So things that may be understandable and common language in your lab may not be so for other people. So have somebody else review it
Great.
outside the lab is also good.
That's a great point. I think we're at time. So thank you all. Please use the LinkedIn chat Q&A that Jason will send out if you have other questions. Please don't forget the deadline, March 1st, for initial abstract submission. And you can find all of the logistics information about the meeting and the submission on our website. Thank you, everyone.